So first, what is a proton gradient? A proton gradient is the product of electron transfer chain wherein there is a higher concentration of protons outside the inner membrane of the mitochondria than inside the membrane, and that is the driving force of the ATP synthesis. As you can see in the picture, the encircled part of the picture is the proton gradient, and they are pumped from the energy released in the ETC. Next, how does the proton gradient lead from the formation of ATP?
The concentration gradient of protons that is generated between the mitochondrial matrix and the intermembrane space provides the energy for the enzyme ATP synthase to phosphorylate ADP, forming ATP. The membrane between the two compartments is very tightly coupled, meaning that it is rare for protons to diffuse directly across the membrane despite the concentration gradient. ATP synthase is one of the few places where protons can travel into the matrix. The movement of protons back into the matrix provides the energy for ATP synthase to combine ADP and inorganic phosphate, forming ATP.
And lastly, what will be happen if the lipid bilayer of the minor membrane is permeable?
If the lipid per if the if the If the lipid bilayer is permeable, the hydrogen atoms in the proton gradient would float helplessly across the membrane, and there won't be much hydrogen ions to go through the ATP synthase complex where ATP is formed.
And that is all there is to know about proton gradient.
Thank you.
Paul D. Boyer, or Paul de los Boyer. Who is Paul D. Boyer? Paul de los Boyer was born on July 31, 1918. He is an American biochemist, analytical chemist, and a professor of chemistry at University of California, Los Angeles. Boyer was born in Provo, Utah. In 2003, he was one of 22 Nobel laureates who signed the Humanities Manifesto. After Boyer received his PhD degree in biochemistry from the University of Wisconsin-Madison in 1943, he spent years at Stanford University on a war-related research project dedicated to stabilization of serum albumin of transfusion. Paul Boyer was associate editor of the Annual Review of Biochemistry from 1963 to 1989. He was editor of the classic series called The Enzymes. In 1955, he received a Guggenheim Fellowship and worked with Professor Hugo Theorel on the mechanism of alcohol dehydrogenase. In 1959 to 1960, he served as chairman of the biochemistry section of the American Chemical Society, and in 1969 to 1970 as president of the American Society of Biological Chemists. Since 1963, he has been a professor of the Department of Chemistry and Biochemistry at University of California, Los Angeles. When does ATP synthase discovered? Boyer began his research on ATP formation in the early 1950s. Scientists know that ATP was the energy carrier of living cells. Adenosine triphosphate consists of molecule of adenosine linked by high energy bond to a chain of 3 phosphate groups.
ATP synthase catalyzes the reaction to combine ADP, or adenosine diphosphate, with a single phosphate unit to yield ATP. The enzyme itself is a complex machine composed of an ion pump and two molecular motors that power the enzyme. The reaction is stimulated when protons are pushed through the cell membrane's proton gradient. The proton gradient is located in the mitochondria. The hydrogen ions present in the gradient act as the source of power for the ATP synthase. and assists in the formation of ATP by adding one phosphate to the adenosine diphosphate already present. ATP is a powerful energy source stored in the nucleoplasm and cytoplasm of cells. The molecule is pertinent for cellular operations to occur. Energy from ATP is released when the molecule loses a phosphate group, which thus forms ADP. The process is then recycled back to ATP with the help of ATP synthase as the ADP molecule passes through the mitochondria. ADP and ATP play an integral role in the production and use of energy. Their cyclical relationship allows the cell's energy to be efficiently restored. ATP synthase is an enzyme that is used to make a compound for the cell that is rich in energy. ATP is often referred to as the energy currency of life. The protein machines that are powered by ATP perform a number of different functions such as the manufacture of RNA, DNA, and protein, cleaning up cellular waste and debris, moving chemicals out of, into, and within cells. The minuscule size of the membranes on which the synthesis occurs dictates that molecules must be manipulated one at a time during the process. ATP is manufactured from two other chemicals, adenosine diphosphate or ADP and phosphate. The energy used to convert ATP into new ATP molecules comes in the form of a hydrogen ion gradient. A positively charged electric current is created when the hydrogen ions stream through the ATP molecules. Examples of the flow of this current is the movement in the chloroplast from the lumen to the chloroplast, or in the mitochondria from the intermembrane space into the matrix.